Let's see. I'm just missing one person. Alrighty. So we are back live streaming again. Yay. All right, and that is mostly for the benefit of those who are absent. Um, we are again in the session of protein synthesis. So we're going to talk about, but before I go to that, I'm going to go back because we can't look at protein synthesis without you understanding. Come on, go back. Your DNA molecule. Okay, so actually, I'm going to go back just a little bit. So we are going to look at this replication, but I have that up in another picture for you to look at in a few minutes. But what I want to go back to before we do that is your DNA molecule, because it's going to, sometimes trying to go back this way is a lot slower than just going and selecting it. There we go. That's what I want. All right. It's important that you get an understanding of complementary base pairing is what it's called. The things in the middle are the bases. So DNA is made up of three things: the phosphate and the sugar, which are the little houses, and the sides of the ladder make up the phosphate and sugar part. But in the middle, we have four bases, four nitrogen bases, and they are complementary. Okay, they're held together by hydrogen bonds. All right, so those of you, if you, by some reason that you haven't turned that in yet, make sure you turn it in. Um, excuse me. But it's important that you remember that these are the four bases in DNA, and they are complementary. So A matches with T always. G matches with C always. The reason these have special shapes is because the complementary pairs fit into each other. They don't fit into things that are not the same. So that's why these shapes, even though the G and the C are repeated in a different order at the bottom, it's still going to be G and C, and they're still going to match each other. All right. Um, so we're going to look at those complementary pairs, and then we're going to look at how we're going to start with a replication, and then we're going to um, go to protein synthesis, because the whole point of um, making a copy of itself is so that it has instructions. Let me see, come back here. Alrighty. All right, so we're going to take this picture and we're going to look at that again because that's just the first step. And the other thing you have to remember is DNA only exists in the nucleus. It never leaves the nucleus, but it has all the information for making the protein. So in order to get that information out of the nucleus, it has to make a carbon copy of itself. So that brings us to today. And that's all right, so that video, I'll make sure that it gets in so on the playlist now that we're back to taping again. All right, so that first part is replication. Then we have two more things that we have to remember as we go through: transcription and translation. Now, unfortunately, both of them start with a T, but you're going to have to remember which one comes first and where the location of it is. So we're going to start off with the amoeba sisters, and then we're going to come back to that. And okay.
After learning about DNA, have you ever wondered how can the DNA actually result in a trait? Let's take an example, like eye color. Yes, your DNA has the genetic information that codes for the color of your eyes. Your eye color is based on a pigment that is inside the eyes, but in order to have that pigment, you have genes, which are portions of DNA that can code for proteins which help make that pigment. So, what we're going to talk about is how your DNA can lead to the making of a protein. This process is called protein synthesis. Synthesis essentially means to make something, so protein synthesis means to make protein. And you may wonder, what's the big deal about proteins? Well, you may not realize this, but proteins are kind of a big deal. They do all kinds of things. Proteins are involved in transport, in structure, in acting as enzymes that make all kinds of materials, in protecting the body, and so much more. You've got to make proteins. It's essential for you to live. And what is so cool is that you are making proteins right now as you sit and watch this video. It's happening in your cells. They're making proteins. So back to your DNA and its role in all of this. All of your cells have DNA. Well, a few exceptions. And that DNA is in the nucleus. Some DNA is non-coding DNA. Some DNA makes up genes that are not activated. More about that in our gene regulation video, but we're going to talk about genes that are coding for active proteins. So how are we going to get the information from these genes out of the nucleus so that the cell can start producing the proteins that it needs to make? Well, let us introduce you to the amazing work of RNA. We have a video comparing and contrasting RNA and DNA. We'll be brief here in saying that RNA is a nucleic acid, like DNA, but it has a few differences. Its role in protein synthesis
Okay,
also
I'm gonna
is
stop right
huge.
there. So first thing you have to remember is, and she'll go through it, but both of those are considered nucleic acids. The DNA has deoxyribose sugar. RNA has ribose sugar. That's what the first initials are stand for. DNA is double-stranded. RNA is single-stranded. But most importantly, DNA has thymine and RNA has uracil. So that's important to remember. Okay. And it's from here on out, most of our conversation will be about RNA because after it makes a copy and leaves the nucleus, we're going to spend the rest of our conversation talking about RNA.
Before we get into the process, please note our typical disclaimer. We tend to simplify topics, but as always, we hope that you have the desire to explore this complex, amazing process later on, to learn all about the extra information that we don't have the ability to include in this short video. In protein synthesis, we can look at two major steps. One is transcription and the other is translation. Transcription has a C in it, and translation has an L in it. I remember that C comes before L in the alphabet, which helps me remember that transcription comes first. I like a lot of alphabet mnemonics. Now, transcription is when we're going to transcribe the DNA into a message. In your cells, the DNA is in the nucleus, so therefore we're doing transcription in the nucleus. In the step of transcription, an enzyme called RNA polymerase will connect complementary RNA bases to the DNA. These RNA bases are bonded together to form a single-stranded mRNA. The M in mRNA stands for messenger. Messenger RNA consists of a message made of RNA that has been based on the DNA. We do want to mention that this mRNA is not usually ready to go right away. There's usually a significant amount of mRNA editing that occurs. We highly encourage you to do some reading about that because it's not only fascinating, it's critical for the process to work correctly. So what's something great about being mRNA? Well, in eukaryotes, you get out of the nucleus. The mRNA can go out of the nucleus into the cytoplasm where it's going to attach to a ribosome. Ribosomes make protein. The ribosome is made of rRNA, and that's an easy one to remember because the R stands for ribosomal RNA. The ribosome is going to build our protein in the next step called translation. You know, you can find a lot of great clips and animations on translation that are just fantastic. We're just going to break down some basics of what's happening. In the cytoplasm, if you look at this, you have all these tRNA molecules available. tRNA stands for transfer RNA. They carry an amino acid on them. An amino acid is the monomer for a protein. It's a building block for protein. Since we're making proteins, we're going to need those amino acids to build it. If you have a bunch of amino acids together, you can build a protein. So it's the tRNA that is going to bring those amino acids together to make that. But wait, how does the tRNA know which amino acids to bring? That's why the mRNA, the message, is so important because it's going to direct which tRNAs come in and therefore which amino acids are transferred. All of these tRNAs are looking for complementary bases. When they find the complementary bases on the mRNA, they transfer their amino acid. When tRNA is bringing in the amino acids, it reads the bases, represented by these letters here on the mRNA, in threes. So it doesn't read one letter at a time, it reads in triplets. That's called a codon. So for example, in this mRNA, the tRNA would read the codon AUG. One of these tRNAs contains a complementary anticodon.
Okay, I'm going to stop it right there for a minute. Let me move that out the way. All right, so it's important that you understand that the transfer RNA already have a triplet code on them because their job is to go and get the amino acid that it's coded for. All amino acids have a what they call a triplet code. There are three bases that code for each amino acid, and there are only 20 amino acids. Like we have 26 letters to the alphabet, there are 20 amino acids, and depending on the uh, arrangement, will determine what kind of protein it's going to make. So each of these is going to figure out only the one of these that matches this AUG or that is complementary to the AUG will actually bind. Okay, and it has an amino acid attached to it.
Which in this case is UAC. All tRNAs that have the anticodon UAC will be carrying an amino acid called methionine. A tRNA with the UAC anticodon comes in to pair with the complementary AUG codon on the mRNA. It transfers the amino acid it carries, methionine. The tRNA will eventually leave, but it will leave behind its amino acid. That's the first amino acid before looking at the next codon. Before we do the next codon to carry this on, if you're wondering, how did you know that the tRNA that went with the AUG codon would be carrying an amino acid called methionine? Well, for that, you will find a codon chart helpful. You can learn to use a codon chart to determine which amino acid each mRNA codon will code for. Isn't it so fascinating that scientists have been able to determine which amino acid corresponds with these codons? I used to have a codon chart poster and just marvel at that. You could see on a codon chart that the AUG codon on the mRNA codes for methionine. AUG is also considered a start codon, as methionine is typically going to be your first amino acid in proteins. There are many types of amino acids in the codon chart, but there are even more possible codon combinations. That means there can be more than one codon that code for the same amino acid. For example, according to the mRNA codon chart, all of these mRNA codons here code for the same amino acid, leucine. That means their complementary tRNAs all carry the same amino acid, leucine. Okay, so going back to the mRNA, let's try the next codon on this mRNA, CCA. On the codon chart, you can see that codes for the amino acid proline. The complementary tRNA has the anticodon GGU. And look, there's the proline that we knew it would be carrying. The tRNA will transfer that amino acid and eventually leave where it can pick up another amino acid. These amino acids are held together by a peptide bond, and it will keep on growing. Typically, at the very end of the mRNA, there is a stop codon. Stop codons do not code for an amino acid, but when the ribosome reaches it, it indicates that the protein building is finished. So the result of translation is that you built a chain of amino acids that were brought in certain sequences based on the coding of the mRNA. But remember, that mRNA was complementary to the DNA, so the DNA ultimately was the director of the entire protein building. Of course, it couldn't have done it without some serious help from mRNA, rRNA, and tRNA. Protein folding and modification may occur, and the protein may need to be transported. This can all vary based on the protein structure and function. Another fascinating topic for another Amoeba Sisters video. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, so I know that was a mouthful, so we're going to break it down. Um, what I want you to do, I need to stop sharing for a minute to do attendance. So I want you to come back here and look this over, um, paying particular attention to this information right here. Okay. And learning the difference between transcription and translation. I'll be right back as soon as I record the attendance. What just happened? Okay. It is helpful when only one person is absent. Okay. So both of those videos have been saved on the playlist since we're back in action here. Okay, let me come back to sharing. All right, so again, there are two words that you have to remember and the order in which they occur. So transcription is when we are copying the information from the DNA. So the same way we looked at replication, the DNA is going to unzip. Oh, the other thing that you need to remember is the monomer. The base units are made up of a phosphate, a sugar, and a base. So all of those, when we get to talking about transfer RNA, those things will be floating around individually so that they can be connected. Okay, so the messenger RNA, oops, They compared it to being a foreign language. So it transcribes the message from the DNA, but then it has to take that message out, and it's like a carbon copy, and it needs to be translated because it's still in a form that you can't understand for making the protein. Okay, and they compared this one to taking that information to that it's a foreign language, and you have to translate it into a language that the amino acids understand. Okay. All right, so let's look at this one, and it's kind of just repeating a little bit of what they did.
Step 1 of protein synthesis, transcription. It is the process of forming a short strand of mRNA from one gene on a long DNA strand. The mRNA strand serves as a disposable photocopy of the master DNA code for a gene locked in the vault, which is the nucleus. We wouldn't want to use our master code, the DNA, to make our protein recipes, right? We use a photocopy mRNA instead. In this stage, we are changing the script of one gene from DNA letters into RNA letters. Step 1 of transcription, initiation. The DNA double helix is separated by RNA polymerase to make a transcription bubble at the start of the gene desired. RNA polymerase begins matching complementary base pairs of RNA nucleotides to the DNA. Note that it matches U with A. RNA does not have thymine. Transcription, Part 2. Elongation. The mRNA sequence elongates as RNA polymerase moves along the DNA. This occurs at a rate of 40 nucleotides per second. Transcription, Part 3. Termination. RNA polymerase reaches a termination sequence and detaches from the DNA. The completed mRNA molecule is released. The result of transcription: a strand of mRNA that carries a copy of DNA, which are the instructions for making proteins. The mRNA has a start and stop codon. A codon is 3 nitrogen bases or letters on mRNA or DNA that carries the code for an amino acid. The mRNA now travels from the nucleus to a ribosome.
All right, let me go back to that picture right there. All right, so this is a picture of what she was just talking about, whereas it's still a triplet code, but that triplet code says this is where we're going to start our copying our information. And then it's going to copy all of the other triplets. And then this last one says, okay, stop copying because we already have the information we need for making that particular protein. Okay. All right, let me. All right, so that's part 1. Now, okay, well, I'm going to repeat it when I go to the picture. So let's look at part 2.
Step 2, translation. The purpose of translation is to make amino acids using the instructions in the mRNA strand. Remember that amino acids have to be made first since they are the building blocks of proteins. When the mRNA strand gets to the ribosome, it is in the wrong language. The mRNA needs to be translated into protein language, amino acids. Step 1 of translation, initiation. mRNA is transported to the cytoplasm and attaches to a ribosome so that the start codon is read first. A tRNA with an anticodon that is complementary to the start codon arrives at the ribosome, donating its amino acid. The tRNA is recycled and is free to pick up another amino acid. Step 2 of translation, elongation. The ribosome moves along the mRNA strand, reading the next codon of the mRNA in order. A new tRNA with an anti-codon that is complementary to the next codon arrives at the ribosome, donating its amino acid. The tRNA is carrying an amino acid that is specific to that codon. This process repeats for an mRNA codon. The chain of amino acids at the ribosomes grow as more codons are read. Step 3 of translation, termination. When the stop codon is reached, translation stops. The mRNA leaves the ribosome. The polypeptide chain will fold into its three-dimensional shape, be taken to the Golgi apparatus where it will be processed, perhaps joined with other polypeptide chains, and packaged to be delivered as a final protein product to wherever it is needed.
Okay, so what I have done for you, we're going to come back to this activity in a minute, but go to the next slide, and what I've done for you is I've put this picture in. All right, so we just need to look at the top picture first, and actually that's very tiny, but I'm going to pull it up as a on my screen, but I put this in here, and we're going to go through what all of these are for, because that's you're going to need to know for your for your activity. So give me just a second to pull up a bigger than that, but you have a copy of it, so that's the important part. Oh, there we go. And I don't think you're seeing that. I don't know why that thing was so loud. She was very loud. All right, let me switch screens. I'm going to stop sharing for a second. You stay where you are. I'm just going to, my picture is on a different screen, so I got to change screens. Okay, there we go. All right, so step 1, the DNA has to open up. And so the first picture is actually the picture that you had of DNA replication, but RNA happens the same way. The DNA has to unzip, and then it creates so it sits there like a template and says, copy me. And then the free floating nucleotide, you see that, but at the top in the right-hand corner, it says free nucleotides. They have to line up. That nucleotide has got a phosphate, a sugar, and a base. So you notice they're different colors, and on the bottom corner, they've color-coded the bases for you. So each one of those is a different type of nucleotide. Now, for this first picture is DNA. If this was raw RNA, those nucleotides, one of those would be uracil. It would not be thymine. So we wouldn't have any. So the things that are going to line up on the template are going to be the bases that are complementary to the ones they see on the DNA, except every time you see an adenine, you're going to get a uracil. Okay, and then they connect to each other. So that she mentioned a bubble, and so that it knows where to start. So because we have this long chain of DNA, and it codes for all kinds of genes, and we're going to talk about that later on in another unit. But right now, just know that a segment of the DNA opens up, and it's going to read just that segment, so it knows what protein to make from that segment. Then, so the second picture in the middle is showing you that it has left that big bubble at the top is the nucleus, and the DNA has opened up, and the little black strip in the middle represents the strand that has been copied, and it has been transcribed. Okay, step 1 is transcription. That transcribed strand of RNA is then going to leave the nucleus. So we have this long strand of RNA, and it goes to the ribosome. Now, the ribosome is like a little bubble, and some of the strand goes through it like a conveyor belt. Okay, so it's going to go through piece at a time. So it looks more like the one on the end here, where pieces of the RNA are going through and three bases at a time are going to be read. Now, you've got the next thing that you have to remember is the transfer RNA. Transfer RNAs already have a triplet code, and they have the little black thing on the end in the middle is an amino acid. And it's a special amino acid to that triplet code. So it reads the conveyor belt, and it says, these are the three I need. So that transfer RNA is going to line up with that triplet code and bring the amino acid it needs to get started. And it stays there until the second one comes, and then the two amino acids will connect. And then the transfer RNA goes away and picks up another amino acid and gets ready to be used again. Okay, so the transfer RNAs come and go, but they only have the unique amino acid attached to it that is necessary to complete the message on the conveyor belt. Okay, so notice at the top, question. Okay, notice on the right side picture, the little green one, that's a ribosome. Go ahead.
Yeah, it's slightly off topic, but I was trying to go ahead and pull up the assignment so I can do it later. But it says the animation or whatever it was is blocked.
Okay, don't worry about that. I've now added it to the your playlist. Yeah, I had put them there as a placeholder so that I would know which video I showed. So don't worry about that one right now. I went back, now that we're back to taping again, because I couldn't even add the videos to the playlist while it was blocked. So now I can add them. I just did. So you should be able to access that video from the playlist. And I guess if you can access it from the playlist, it shouldn't be blocked, right? As long as it's on your playlist. Is that how that works? Yeah. Okay, but I did put it there. All right, so I put both, and the ones that we watched today, I put that on your playlist as well. So both of those have been saved. All right, so again, the important thing is that the transfer RNA is going to go away, and the little multicolored chain at the top, that is our protein. Okay, think of a protein as a string of pearls, and each bead on the pearl is a different amino acid. Okay, and the arrangement of the amino acids, the same way your name is spelled with certain letters, and it's your name, and so when you see your name on paper, we know to identify it with you. When we look at the sequence of amino acids, we know to identify it with a particular protein. Okay, all right. So again, I have this picture in your thingy there. Let me come, I'm gonna have to come out, stop sharing and come back. So this is just blown up. I thought it was more detailed than the one they have in Canvas. Okay. So let's get back to that one. Let me close this. All right, there we go. All right. All right. So what we're going to do, so yeah, this video that's here is the one we just watched. So don't worry about that. Now that we're back to taping, I might even take that off, but I can leave it there. All right, in the video, she talked about reading a chart, a triplet code chart. So this is, I've enlarged this for you, but it's also on the worksheet. And how this works, let me change this so I can make sure that you're looking at what I'm looking at. Okay, so this is a codon chart. And you see where it says first letter, second letter, third letter. So you actually have to go around the box in order to use this chart. So again, our little conveyor belt has three amino acids, three bases set up, and it's telling us what amino acid to go for. So if, let's say, okay, let's do CCG. Just pick random letters. All right, so if I'm going CCG are the bases on my messenger RNA. The transfer RNA is going to be the one that has the complementary code to it. So what I'm looking at, C is the first letter. So I start on this side, and that tells me the amino acid I'm looking for is in the second row. Okay, then I go up to the top. But as you see, you've got 4 columns in the second row. Then I go up to the top and look at the second C. That second C is in the second column. So I'm going to come down to the second row in the second column. And then the third letter is G. So I'm looking on the side over here. G is going to be my bottom row of that box. Okay, so so when I get there, it's CCG is the bottom one in the second row, second box, and PRO stands for proline. Okay, these are all been abbreviated, but it has the name of proline. So that's the amino acid that's going to be retrieved when we try to build our protein. All right, notice that this one down here in red says AUG. She mentioned that in the video. They always know where to start. Methionine is always going to be the first amino acid because it tells it where to start. Then you come up here to the top corner and you've got 3 different messages on how to stop. You've got UAA, UAG, and UGA, not the university. Okay, and they don't code for an amino acid; it codes for stop. Don't make any more protein. Okay, so the first one does code for the amino acid methionine, so it's always going to be the first one. But then these tell you when to stop. So when you see that, that means stop. All right, there's a second way to do the, and it's a wheel. And I find the wheel is slightly more complicated than the box, but it works the same way. You start on the outside. Actually, you start on the inside and move your way out. So same thing, if I said CCG, I would start with that C in the corner. So that's going to tell me that everything I need is in the bottom triangle of that pie. Okay, it's in that C section of the pie. And then the second one is C. So I come out to the second ring and I find that C. And then I come to the third ring and I'm looking for the G. So you still got 4 different letters in that third ring, and it's still going to tell me that my amino acid is proline. Okay, so there's 2 different charts that you can use to get to the same place. Like I said, I find the box a lot easier to understand, but this one, you might see this on a test, so you need to be familiar with how to use it. Okay, so I'll go through that one again. If I used the example of CCG, you start in the middle, and the C tells me I'm working with just that part of the pie that's at the bottom right-hand side. Then I go to the second ring, and that second C, and then I go so that gives me another 4 letters to work with, and all 4 of those letters just happen to go for the same amino acid. So it didn't matter whether it was U, C, A, or G, it was still going to be proline. And she mentioned that in the video too, that some amino acids have 4 ways or 3 ways that you can code for that same amino acid. Okay. Alrighty, that takes us to any questions so far? Questions. All right, so go to your next slide, and that's where the assignment is. And you're going to practice, practice, practice. Uh-oh. Well, maybe yours will work. Mine is still loading. Are you getting that message too, that it's not loaded? Or is yours pulling up? I may have to upload this again. Mm, there it is. It just took a minute. All right. So at the top, it gives you a little review of what protein synthesis is, and it's the process in DNA that builds the proteins from amino acids. So again, the whole point of this is to get those little beads to line up in the correct order to make the protein that is needed for whatever it is it's trying to make. It could be making skin, it could be making eyes, but you need the protein for to be coded for that. Also, she kept talking, they kept using it, it changes languages, not speaking the same language. Think of it more like when the messenger RNA is copying the DNA, it creates a reverse copy. Okay, so it's backwards. So when it goes to the ribosome, then the translation translates it back into the original version. Okay, so you guys like getting a carbon copy and then printing it back to what the original looked like again. So the carbon copy is backwards, and then the transfer RNA transfers. So the transfer RNA chain is going to look more like what the DNA looks like, except it's still going to have uracil because it is still RNA. Okay, all right. So we do transcription first from the DNA to messenger RNA. Messenger RNA then takes it out and goes to the ribosome. And again, ribosomes are those little bubbles. So the whole strand does not fit inside the factory. Okay, we called the ribosome the protein factory. The whole strip doesn't go fit in the factory. It is read three bases at a time. And it goes through like a little ticker tape. Okay. All right. You have to remember that it takes 3 bases to make a codon. And the codon signals 1 amino acid. And then we want to put all the amino acids together to make a chain, and that's a protein. Okay. All right. So then here's again a copy of the chart, both charts for you to use, and then you got some practice to do. All right. So you're going to take each one of those letters, you're going to go up to the chart. So I'm going to do the first one with you. It says it what? What is this? The worksheet is locked? What is locked? Yep. Ah, how is that locked? How do you lock a worksheet? Okay, let me finish. Oh no, let me stop and try to. Okay, give me a minute. I'm gonna stop sharing. And it might be like how it would. Oh, you know why? Because I shared it from the other class. So let me go back. The 13th of today's the 12th. Okie dokie. Thank you for clearing that up. All right, today is the 12th. Easy fix. I like when things are easily fixed. There we go. There we go. All right, so I have changed the date to today and not the 13th, which is tomorrow. I don't see you tomorrow. Okay. All right, so try it now. I'm having that kind of a day. All right. Are you able to get into it now? Okay, so I'm going to do the first one with you. And again, the way you use the chart, see where it says first base, second base, third base. You have 3 letters. So that's how you want to read it. So the first one, just like our example I gave earlier, is C. So you want to find C on the side that says first base. Second letter, so that's telling me that the answer that I'm looking for is in the second row. Then the second letter, at the second letter, I go up to the top where it says second base, and that's a C also. So now it's telling me, so I come down to the second row, second column, and that's where my answer is going to be. Now, like I said before, it just so happens that the third letter is also C, so you can see that there. If the third letter is C, our first one is proline. Okay, now all of these have been abbreviated, so it's okay if you write PRO on the line, but PRO is short for proline. So that's what you want to write there. Let me go over, I'm going on the other side to the AGU. Okay, AGU. So my first letter is A, so that tells me it's in the third row. My second letter is G, so when I come up to the top, that's the 4th column. So now I want to come down from my 4th column to my 3rd row, and the last letter is U. So I find U on the 3rd, and that's the top one. And in this case, you do have 2 different amino acids. So I want the top one at the top, AGU, and it's serine. Serine. But again, it's okay for you to write SER on your line. All right. Anybody need me to do one more? You got it? You don't have it. Do one more. Okay. Coming back to the other side. CAG. So I'm looking at that one. CAG. So, and again, follow the words on the side of the box. It says first base and codon. My first base and codon is a G, so I look for the G. And that's the bottom one on that side that says first base. So that means the answer has to be in that 4th row at the bottom. The second codon is, I'm sorry, what am I looking at? I was supposed to be doing CAG, right? I was looking at all right, let me go back up here. CAG. So C is, again, it's going to be the 2nd row. And then the second letter is A. So at the second base, A is the 3rd column. So I'm going to go from my 3rd column down to my 2nd row because that's where the C was. And then the 3rd base is G. So if I'm looking at run my finger from the G on the 3rd side, that's the bottom ones. So CAG is the abbreviation is for GIN. That's what you'll write answer on your line. The amino acid is GIN. All right, so I'm not asking you to learn all the names of all of these. It's okay if you just use the chart, know the abbreviation. Okay, so this is the practice. Now, all we're doing right now is identifying amino acids. When you get to the second part, see down here, they have some of them written out in full. Down here, they want you to go back to the chart find the initials, and that, so they've highlighted the first three initials here. Go back up here and figure out what would be the three letters that would code for that amino acid. So up here, you're practicing finding the name of the amino acid. For number two, they've given you the name of the amino acid. Go back and tell me what would be the codon for it. What are the three letters that would code for that amino acid? Okay, so again, proline is our first one. So we went up here and it was the one that we had used earlier. So I find PRO on the chart and I have several possibilities. So for this one, I need you to write all the possibilities if it has more than one. All right, so that answer, I need you to write, it could be CCU, it could be CCC, it could be CCA, or it could be CCG. So that's what you have to do for these down here. If it has more than one, some of them have two, some of them have four. If it has more than one, show all the possible codons that it could possibly be. All right, then down here, again, on the chart, it says start. So that's the one that you want to write over here for start. The three colons for stop. There are three of them, like I pointed out before, and they're highlighted for you also. So those are stops. So you need to write all three of those on the line for that one. And then this one is where you are actually going to take that chain of amino acid, and they've already set it up in groups of three. You are going to first translate and make a messenger RNA, and then you're going to translate that messenger RNA so that you have the correct protein. Okay? And remember, every time you have an A on an RNA, it's going to be a U. It's not going to be T. All right. Any questions? Any questions? All right, we have a few minutes. So you can just get started with the top and we'll review this again next class and get you finished up with this worksheet. So this is making a protein. All right, everybody good? Any questions? Any questions? All right, don't worry about the number 5 today. I'm going to go practice that with you again next class. But you can get through 1 through 4. Okay, so do at least 1 through 4 right now, and then we'll worry about number 5 next class. All right, we got about 10 minutes. And again, if the chart on the worksheet is too small, I blew it up for you in the lesson box in the previous one. All right, so that it's a little bigger there. And on this one, it also shows you in green that the methionine is the start, and all the little red squares are the stop codons. All right, now this is just reference. This is not an assignment, so there's no grade associated with this one, but there is with the worksheet. Why do you have to have? All right, so again, don't try to submit this worksheet yet. Wait till Friday, and we'll talk about the question number 5. All right, Imani, check my comment on the graded assignment. All right, and again, if you haven't submitted your DNA replication today, it's due today. So that means after today, it's late. So make sure that you get that turned in. There's about 4 of you who have not turned that in at all. Again, the DNA replication worksheet has a due date of today. So make sure that you work on that first. Don't worry about this DNA protein synthesis. Make sure you finish your DNA replication worksheet first. What is important? Okay, folks, time is up. Again, it's important that you get that first worksheet done because today is the due date. So tomorrow it'll be late. You all have a great rest of your day and see you on Friday. And we'll talk some more about how to do that DNA, create that protein chain. Imani, compare your diagram to the one that's in Canvas, and that one is properly labeled as well. All right, bye, folks.